Okay, in this video we're going to talk about mutations. So mutations, all that is a change in the DNA. Now in the last unit we talked about how DNA codes for proteins. So remember we transcribe it to the mRNA and then we translate it into a protein. So if we change the DNA, eventually that could lead to a change in the protein. There are lots of causes of DNA. So the causes could be radiation, like X-rays, UV light from the sun. It could also be things like smoking, just other chemicals, or alcohol. So all of these things are causes of mutations. Mutations are most likely to occur during, or when we're copying that DNA, so during replication. So most likely to happen it's most likely to happen during replication. Remember that's going to be during the S phase of interphase. And so yeah, that is when it's most likely to happen. Alright, so there's 6 examples of mutations that we're going to talk about. The first one is substitution. Right, and our original strand, I'm going to write it right here in green. The original strand is going to be A, T, C, C, G. Alright, and so in substitution, it's just like during a sport. So if the coach substitutes a player, one player goes out and another one goes in. So we are going to swap an A for a T. So it's going to be A, T, T, C, C, G. Now notice we always read 3 letters at a time. So we're going to read these 3 and these 3, because 3 letters is what codes for the amino acid after we transcribe it to the messenger RNA. So this right here, the A is out, and now that T it was put in. So just like in a sports game, when you substitute a player, one goes out, one goes in. This should have been an A, but now it's a T. All right, second example is insertion. Okay, and so that is just when you insert a letter. So one example would be is if we inserted another A. So remember, we always look at our original strand. It was supposed to be AAT, but instead it's going to be AAA, T, C, C, G. So what happened was I added this extra A. Now what that does is that changes the frame that we read. So instead of AAT, now the first one's going to be AAA. And it changes every single, it's going to change every single amino acid after that. So that's really bad. We're going to come back to that in just a second. The next one would be a deletion. And for a deletion, it's just like it sounds. You're going to delete something. So we're going to go ahead, think about what I am deleting. Okay, so if we compare to the original strand, you can see that that T is not there anymore. So this T was deleted. All right, and once again, that's instead of being AAT, which is going to eventually code for a protein or an amino acid, it's going to read AAC, and then we're a short one, CG. All right, so we're going to pause after those first three. So remember, substitution, you're substituting just like if you're playing a football game or basketball game. If one player goes out, one goes in. So the T was put in and that A was taken out. This is also called a point mutation. And that's because only one point is changing. None of the other amino acids are going to change down the line. Insertions and deletions, though, these are called frame shift mutations. And these are really bad because normally we read 3 letters at a time. So it should be AAT. However, because we added in this extra A for the insertion, we inserted an extra A, it's going to change all of the 3's. So instead of AAT, we have AAA. Instead of CCG, we have TCC. And it's going to switch all of them after that. And what happens is because we inserted an extra one, everything gets pushed over to the right. So these are really, really bad. Same thing with the deletion. Because we, remember we had AAT originally, now we have AAC because that T was taken out, everything gets pushed to the left. And so whatever was after this, everything's going to slide to the left. And it changes the reading frame. It shifts the whole amino acid. And usually these aren't going to work now. All right. So point mutation is same thing as substitution, and then frame shifts. These are really bad, and this could be insertion or deletion because it changes all of the amino acids, not just one. All right. Then the next few that we're going to talk about, number 4, is an inversion. And so this is not insertion, but inversion with a V. So the way I tell mine to remember this, you have your V, and all you're going to do is flip it upside down. Okay, so you have the V, and you're just going to flip it. And so to invert just means to flip. So instead of AAT, CCG, we are going to flip some of those letters. So we're going to actually flip this middle 4. So it's going to be A, C, C, T, A, G. So right here, this middle 4 is flipped backwards. So A, T, C, C was flipped. And you can see, so that's going to change these 2 amino acids, but it's not going to change any of the ones after that. So yep, inversion just means to flip. All right, moving on. Next one is duplication. Okay, and a duplication just means to make an extra copy or to double. So copy or double. And so you just accidentally double something. So instead of A, T, C, C, G, let's say we have A, T, which is normal, and then let's say we accidentally double that AT again, and then C, C, G. So you can see this part, the AT is not supposed to be in there. We accidentally just doubled this part and did it twice on accident. So duplication, you just accidentally double something. It is similar to an insertion. However, duplication could be duplicating a big piece of a chromosome and not just a few bases. So you could also see it. You could see that a couple different ways. But look at the sock credit for that. I've included another example. All right, and then the last one is translocation. And so with translocation, this happens on a chromosome. So let's say I have 2 chromosomes. Here's going to be one chromosome. And here is another chromosome. Alright, and what is going to happen is, for translocation, I want you to think of like a transplant. So they're going to be switching parts to them. So like with the transplant, you give someone a body part, I guess, kind of. You have like a kidney transplant. Then you're going to get a kidney from somebody else. Well, in translocation, they're going to swap parts. So we have the top of the green one, but then a little piece of the purple is going to move over here. And then we have the top of that purple like normal, but then it's going to get the bottom of the green. And so what happened was the chromosomes switched parts. And remember, this was not supposed to happen. Alright, and that pretty much wraps up mutations. Once again, to see a duplication example on a chromosome, just go and do the practice Socrative.